this evening. So I'm Sebastian Lay. I'm MBL's Chief Advancement Officer, and aside from telling you to silence your cell phones, I'd also like to remind you just at the end of the talk, there will hopefully be some time for a Q&A, but if you don't get to ask a question, there is a reception in the MEGS room in Swope that we'd hope that you join us for, just to continue the conversation. And then my final two things are a little more personal, I'd say. One is to just let you know that Rudolf Oldenbourg, who's a resident faculty member here at the MBL and a major player in our imaging efforts, will be introducing Amy shortly. But I also want to take the opportunity in front of all of you to just personally thank Amy on behalf of the MBL. And really, it'll become apparent, hopefully, in the next few years on the MBL community for all of her help in co-chairing our imaging steering committee. So the MBL has a real strategic focus on developing our imaging research and programs going into the future, building on the strengths that we've developed over the history of this institution. It's a niche that we think we're well poised to deliver on. We've made some real advances in that space just in the last couple of weeks, and that's in no small part thanks to Amy's guidance and a number of other folks who are in the room tonight, who I won't list by name, but all of you who know you are involved in this and continue to advise us and help us. Thank you so much, because it's a really important step for the institution. So thank you, and you all.
Yeah, thank you, Sebastian, and welcome. I have here, my next guest needs no introduction. Well, for many of the MBL community, Amy Gladfelter needs no introduction. For those who want to learn more about Amy's distinguished career path, I refer you to the one-page summary that is available at the entrance. I only mention that in 2016, Amy became a Howard Hughes Medical Institute faculty scholar, an honor that she shares with another Friday evening lecturer this year, namely Manu Prakash. My task here is to tell a few personal observations about Amy and set the stage for the subject she will be talking about. Amy first came to the MBL as a student in the physiology course in 1998. 10 years later, Tim Mitchison, a scientific matchmaker par excellence, brought Amy and the MBL together again. Tim saw a match between the dynamics of higher-order septin structures studied by Amy and the polarized light microscopy techniques that we have been developing in my lab. You might say the relationship between Amy and the MBL is built on mutual attraction. Following the first extended stay in the summer of 2009, Amy and her family, her husband, Mark Borsak, who is affiliated with the Woods Hole Oceanographic Institution, and their children, Sebastian and Stella, have spent summers here in Woods Hole ever since. To do its part, most every year, MBL bestowed a summer fellowship on Amy to make sure she would come back and so enrich the Woods Hole community. Amy was instrumental in supporting and guiding the development of polarized fluorescence microscopy in my lab and in the lab of Tomomi Tani here at the MBL. Her clever experimental designs take maximum advantage of quantitative image data, which she analyzes using physical models to reduce complex image data and reveal hidden symmetries, such as the pairing of septin molecules and its role in membrane binding. Amy not only collaborated with many of the resident and Whitman scientists at MBL, she also contributed to, and when necessary, insisted on the upkeep of services supporting young families of visiting scientists who play such an important role in the vibrancy of this place. This fact couldn't be better illustrated than in the following picture that shows Amy and her young daughter Stella peering down a microscope. The overarching theme of Amy's research is how cells are organized in time and space. Specifically, this theme includes how cells build large, highly organized and dynamic structures using the cytoskeleton. It also includes the variability and yet precision found in cell cycle timing. And more recently, the crowding behavior on Manhattan sidewalks during rush hour. She uses this metaphor very aptly in her abstract to her talk, as people rushing on sidewalks form intermittently stable groups, such as when they meet by chance, stay together and exchange gossip, or because they have to wait together before a traffic light. In cells, these groups or compartments contain specific protein and RNA molecules that together separate into a liquid phase that is distinct from the rest of the cytosol. The condensates are stable, yet rapidly exchanging individual molecules because they are not bounded by a membrane. Amy's talk will apply this new concept of liquid-liquid phase separation to the organization of the cytoplasm in fungi and ultimately to shed light on the formation of plaques and aggregates that are disease agents in neurodegenerative diseases. I'm so proud of Amy and so lucky to be able to work with her. By listening and experiencing Amy's lecture, I trust that you will feel the excitement, the deep knowledge and commitment that she brings to all her endeavors, including finding answers to the question, what do fungi tell us about neurodegenerative disease? Please join me in welcoming Amy to the podium.
Robust hug just dislodged my microphone. There we go. Which is very appropriate. Okay, perfect. Thank you, Rudolf, for that really warm and personal introduction. I couldn't ask for a nicer introduction. It is an absolute pleasure to be here sharing my science with this community that has supported me and our work for 20 years now. So thank you. I want to begin tonight with one of my favorite quotes, and it's about 2,000 years old, written by Pliny the Elder, and I believe it's probably one of the oldest rationalizations for using model organisms to study biology. And so what Pliny wrote to us is that nature is to be found in her entirety nowhere more than in her smallest creatures. And what I hope you take from this talk tonight is an appreciation that the simplest systems can sometimes make the biggest impact in solving problems for very complex systems like us. So I am fascinated by fungi, and you can see one of the organisms I've spent much of my career studying right here. This is a budding yeast. This is what brings us bread and beer and wine. It's also informed us about many of the basic ways that cells grow and divide, and helped us to understand a whole myriad of different human diseases. What you may not fully appreciate, though, is that fungi are everywhere. They absolutely shape and contribute to the terrestrial biosphere as we know it. And while many cells look like these nice, cute, round yeast cells, there are many more fungi out there that grow in these very elaborate, beautiful cells like I'm showing here in this time-lapse movie. And these are a common form of fungi where they grow in these long interconnected branches that can communicate over very long distances. And you're probably familiar with the sort of macroscopic form of fungi in the forest, these amazing fruiting bodies of mushrooms. But what lays beneath these are, in fact, these interconnected cellular highways upon which chemical information is being exchanged over meters and even tens or hundreds of meters. And for this reason, fungi have actually been called sort of the internet of the forest. And what's fascinated me about fungi is this exquisite spatial organization. So fungi are actually masters of multitasking and spatial organization. So what you're looking at here is a time-lapse movie of one of my favorite organisms. It's called Ashbia, and it's a fungus we study in my lab. And it's growing under the microscope, and we've labeled individual nuclei in green. And you can see that this one big cell actually has many copies of its genome in each of these nuclei. And when the nuclei divide, they are false-colored blue. So hopefully what you can see is flashes of blue interspersed throughout time and space within these cells. You can also see that growth is highly focused within these very specific locations. So what's really remarkable about fungi is their ability to spatially control and coordinate all of these different tasks over many different length scales. And this makes them extremely powerful systems for studying patterning in the cytosol. And so what we're going to talk about tonight is both how cells normally spatially organize the cytosol, and we're going to relate this to what goes wrong when cytosol organization goes awry. And essentially what happens is protein aggregation, which is at the root of a whole myriad of many different neurodegenerative diseases, including Alzheimer's, Parkinson's, and ALS. But before we talk about that, let's think about what is cytosol. This is a word that might not mean something to everyone here. So for a moment, close your eyes and just imagine a textbook version of a cell. I can see who's got their eyes open. It's like church. So, okay, it looks something like this, right? So you can imagine there's a container. It's filled with sort of blobs that have membranes around them, things that look like mitochondria, and endoplasmic reticulum, nucleus, all these words that you learn. But the cytosol is all of this essentially empty space, and it's really been neglected in terms of study and understanding. And it's usually depicted just as like water that holds these more important sort of busy parts of the cell. But in reality, the cytosol feels much more like Stony Beach in July. It's a place where molecules are jam-packed together. They're bumping into each other. It's an extraordinarily crowded environment. It's also, as Rudolf alluded to in the introduction, an incredibly dynamic environment. So molecules are moving around and changing places, either through passive diffusion or active transport. And somehow, in this sort of apparent chaos of motion, just like what we see in a city street, molecules end up where they need to do their job. So how does order emerge from this apparent chaos in the cell? So we know a lot about how order can arise through containment of biochemistry in compartments for these sort of conventional membrane-bound organelles that we're all familiar with. But the question is, how does order and organization arise within this vast, teeming wilderness that's jam-packed with molecules? So we know now, because of very recent work, that one way that organization arises in this area of the cytosol is because parts of this area, molecules in the cytosol, can actually condense into liquid-like droplets. And you're seeing one of the first movies that observed this sort of behavior in the cytosol right here. And hopefully what you can see is these compartments actually have very interesting properties. They're liquid-like, they flow, they fuse. They're sort of a lot like how a lava lamp looks if you watch it for a while. So these are very different than the kind of compartments that everyone's sort of familiar with that are contained by membrane. And the way we think about these is as biological condensates. And the idea here is that molecules can organize through the process of a phase separation. So just like water can be in a gas or sort of soluble state, a liquid-like state, such as dew condensing on grass, or a solid-like state in the form of ice, which none of us want to think about on the day after the solstice. It's the same for cellular molecules in the cytoplasm and also in the nucleoplasm. They can be in a more dispersed state, a liquid-like state, like I just showed you in that movie, or in more solid or aggregated states. And so the idea here is that organization is coming about through the material properties of these molecules as they come together. And so what this means is that here we have order again arising through containment in compartments, but in this case, these are biological condensates. These are compartments that form without any lipid bilayers. And so what that means for a cell is that it can actually rapidly assemble or disassemble these compartments, and they can be very dynamic. And molecules can enter and leave in ways that is much more flexible and dynamic than necessarily a membrane-bound compartment. So this is, again, order through compartmentalization, but yet it's a totally different kind of compartment than what we're familiar with. And so it's not by chance I'm talking about these here in the Friday evening lecture, and that's because the MBL played a critical role in the discovery of these compartments and also laying the foundation in a brand new field of cell biology. And so this discovery of condensation as a means for cell organization happened at MBL. And these very first observations of liquid-like compartments happened within this very simple nematode. So Pliny would be very pleased. And essentially, it involved magic that happens especially here, if not only here, through the commingling of diverse scientists. And in this case, it involved the mixing of physicists, biologists, worms, and microscopes within the physiology course. And I want to highlight this observation from Cliff Brangwyn when he was a young physicist. He was a postdoc in Tony Hyman's lab when he observed this sort of behavior within a cell. And with the physicist's perspective, he noticed that this was a really interesting material property of cellular compartments, that they could essentially behave as liquids and have this flowing and fusing behavior. So this was an observation that was born out of the educational environment and this amazing interdisciplinary environment that we have here at the MBL in the summer. And this continued to be fostered here through the last 5 summers in a group called the Summer Institute that was spearheaded by Ron Vale and Mike Rosen and others. And this was a dynamic group where at any given time there were 20 to 25 scientists in row grappling with how do we think about this brand new field? How do we think about these problems? And so this is a picture of us in action from a few summers ago. And I can't say enough about how important it was for the MBL to provide an incubator for this brand new field. So we now know that biological condensation is a nearly universal feature of cells, and it serves many purposes. So we know that condensates form in neurons for the packaging of molecules for long-range transport. We know that most cells will form condensates in response to stress signals. And for example, embryos will take advantage of condensation for patterning and developing body axis plans. So how do condensates actually form? So it involves the interactions of molecules that have these sticky spots, or these areas that allow them to interact either with like molecules or with different molecules. And when you have a whole lot of these sticky molecules together that can interact, this leads to the condensation process. So on the microscopic scale, what's assembling is this meshwork of molecules. It's highly dynamic. It's a liquid. The molecules are changing places. They're rearranging, but they're creating essentially a protected environment that's distinct. It's demixed from the surrounding cellular milieu. And at the macroscopic scale, it leads to the formation of a droplet, which has these liquid properties we saw in the movie. It's got a surface tension, and it's got particular viscosity or gooiness to it. And so what we're seeing specifically are particular kinds of proteins are able to undergo this condensation process. Very commonly, the proteins that do this have features such as a disordered structure, which sounds like an oxymoron because it is. This is proteins that don't take a particular form, but they're highly unstructured and highly dynamic. They're also called prion-like. They share features with prion molecules. And they're often hooked up to a part of a domain that allows the protein to interact with RNA. I just remind everybody that RNA, or messenger RNA, is what links DNA to protein. It conveys that information. And cells are full of RNA. And so in many cases, what we can see is by mixing a protein that has a disordered structure with this ability to bind RNA with RNA itself, we can produce this beautiful liquid-liquid phase separation, or the demixing of these molecules in the absence of any other input. So we don't need to add energy, we don't need to add any other cell components. This happens spontaneously. So what this means is this is essentially a very primitive form of self-organization that cells are able to take advantage of, because we can have this occur just spontaneously in a test tube. So this is occurring simply with two components. So what are these condensates actually doing? So we don't really know. So this is really what everybody is trying to understand. We have many. They are thought to bring factors together to interact at just the right time and just the right place. We think they're serving as vessels for chemical reactions, sequestering them away from the surrounding environment, and maybe actually enhancing the rates of enzymes by putting them into these crowded droplet environments. And finally, there's a lot of interest in them acting as stress sensors of either the temperature or the pH that cells may be experiencing, because they assemble. This condensation is this fundamental physical chemical process. The idea that these could actually serve as basic sensors of the environment is very attractive, and there's some evidence for that. So why might this matter to you? Hopefully you already think it's really interesting, but it may also be relevant to your lives or your family's lives. That's because it relates very directly to protein aggregation, which is a hallmark of many neurodegenerative diseases. And so we're just looking at the stained aggregates from patients that have suffered from Alzheimer's disease or Parkinson's disease or Lou Gehrig's disease, ALS. And so what's becoming increasingly clear is that neurodegenerative diseases such as ALS or frontotemporal dementia, FTD, are likely arising from aberrant forms of condensation. And so the evidence for this first comes from the mutations that have been mapped in patients that suffer from these diseases. And many of these mutations arise in RNA binding proteins. So these are proteins that have both a disordered structure and this ability to interact with RNA. And so what the current hypothesis is for what's going on is that these proteins are normally able to form these liquid condensates in cells, but upon stress or mutations or age, they actually transition into the irreversible neurotoxic aggregates that are a hallmark of many of these neurodegenerative diseases. So far, what's the evidence for this? So work from Tony Hyman's lab looking at a protein called FUS, which is one of the proteins that's found to be mutated in ALS patients, has shown that this protein can readily undergo a liquid-liquid phase separation and form liquid droplets. But if these droplets are allowed to age, or if they're made with proteins that have mutations, such as what are found in patients, you see this striking formation of aggregates or fibers emerging from within the droplets. And so what we can see is that by having the patient mutations present, it actually accelerates the formation of aggregates or fibers from a liquid-like state. Similarly, it was very recently shown with a protein called tau that has for a long time been appreciated as a component of aggregates within Alzheimer's disease. We can see that the human protein tau also can undergo this liquid-liquid phase separation, but in conditions that promote disease, such as mutation or other cellular stresses, we can see that actually aggregates are emerging from these liquid droplets. And so the idea here is that potentially these liquid-like droplets that serve a normal function in cells are actually the birthplace or the birth site, the crucible of fibers that are pathological in these diseases, such as Alzheimer's disease. And so here's a simple model of this, where in a healthy neuron, there's a nice soluble or sort of gaseous pool of the tau protein, or it's forming condensates that are very dynamic. However, as a cell is transitioning through a more pathological state, we see a change in the physical properties of these droplets. They become more viscous or more structured. And then from here, the aggregates are born that ultimately will give rise to the pathology of this disease. And I should say this is not unique to neurons. So in droplets that we take from fungal cells using fungal proteins, we can see the exact same thing. Here are droplets that are made from a fungal protein in RNA, and with time, we readily see the emergence of fibers or aggregates within these, suggesting that this is really a fundamental property of this form of cellular organization. And so I just want to remind everybody of Pliny that this fundamental behavior is often present in even the simplest of cells. So why is it that neurons are so sensitive to these aggregation-prone pathologies? I think there's a lot of hypotheses and ideas about this, but one thing I would like to propose is that it comes partly because of their cell architecture. Neurons are big branched cells that are forming large interconnected networks that have to transmit information over long distances. And they are highly reliant on condensation to package RNAs and proteins for transport over long distances to get them to the right place in the right time. And so these are cells that are highly reliant on this process of spatial organization in the cytosol. And similarly, the cells that I'm most passionate about, fungal cells, are also have a similar architecture. They're very big, they're very branched, and they also need to form interconnected networks through which information is passaged. And this is this ancient function of this shape of this shaped cell. And work from our lab has shown that as a consequence of this cell architecture, that these cells are also highly reliant on condensation for cellular organization. And what we think is going on is that condensed droplets are essentially creating neighborhoods in these big syncytial cells. And syncytia, I want to define, as just a big cell that has lots of nuclei in it, like the fungi I'm showing you. And that by forming liquid droplets in particular areas, it makes this very big cell actually compartmentalized into smaller functional neighborhoods. And you can see that really nicely here in this time-lapse movie. I'm showing you again for the second time so you can really enjoy it. Again, here in this movie, what we're able to see is evidence of cytosolic neighborhoods. And the evidence of these come first in these flashes of blue. And what you can see is that they're in areas of cells where the other nuclei are green, they're not dividing. So the division process is actually quite local. It's actually happening within a specific territory of the cytoplasm. Similarly, growth is highly focused, so we can see it's not happening all across the cell, but the cell is specifically marking and determining particular locations in which it's growing. And so this also requires a regionalization or territory of cytosol to be established. And what's really neat is we can actually see evidence of these territories when we try to measure just the physical properties of the cytosol. And the way we're doing this is taking advantage of little probes that we can follow the diffusion of within the cytoplasm that were developed by Liam Holt, who's here in the audience. And by tracking these probes, we can see whether they're able to move freely and diffuse quickly, or whether they get caught and move much more slowly. And from this, we can infer actually that there's patterning in the cytosol in these cells. And you can see this patterning in this movie in the form of these red spots, which are much stickier than the spots that are blue or green. And what we've been able to show is that these red spots correspond to the neighborhoods. They correspond to the neighborhoods around nuclei and to the sites of growth in the cells. And it turns out they are dependent on the condensation of both proteins and RNAs. So these neighborhoods, these territories are being established by condensed droplets in the vicinity of nuclei, which are these black holes in this movie. Or you can also see droplets condensing at sites where the cell is growing. So these are really dynamic, they're fusing, they're showing all the hallmarks of a liquid droplet in the cell. So what are they made of? They're made of a protein that we call We3, which isn't that important. What's important is that it's got those sticky patches that give us this ability to undergo a liquid-liquid phase separation. So it's got an RNA binding domain, which allows it to interact with RNA molecules. And it's also got a very interesting disordered sticky track, which is called a poly-Q track or polyglutamine track. And all that means is basically it's a run of amino acids, which is what makes up proteins, where you have a whole bunch of glutamine over and over and over again. So it's just a repetitive track of the same amino acid. And this poly-Q track has actually been famous for a very long time, not because of this protein, but because of a protein called Huntington, which is the causative agent of Huntington's disease. And so in the Huntington protein, there is a poly-Q track in the first exon of this protein. And it's been known now for many years that the length of this track is absolutely critical for onset of the disease, so that individuals that have less than 35 of these glutamines in a row remain unaffected. But if this track expands to having more glutamines in it through mutations in DNA replication, we see an expansion, and we can see if you go above 35 that then you see this associate with the pathology. And so we've known for a long time about this being present in proteins like Huntington. What we've been able to show studying this in fungi is that there's really normal physiological roles for these sequences. They're not there just to wreak havoc with our neurons. They're actually quite ancient ways of organizing. And so here's an example where in the normal fungal cells, they grow with this beautiful branching. When we take out that poly-Q track from this protein, they're no longer able to branch. They're no longer able to explore and connect in their environment. And why is that? We know that the sticky poly-Q track is required for this protein to actually condense into droplets. And so here we can see what this protein will do when it's intact completely. When we delete this track, we can see now that this no longer is able to form these liquid-like droplets. And so the reason why it's no longer able to function in the cell is because it can no longer condense. And similarly, very recently, within the last month, a similar story has been shown for the Huntington protein, where depending on the poly-Q track length, the protein can exist in either a soluble or diffuse state, a liquid-like state, or as the track gets longer, it transitions into the solid, fibrous, aggregated state here. So I want to remind you, though, that many of the neurodegenerative diseases that I mentioned actually arise from mutations in RNA binding proteins. So why is RNA interaction such a common feature in these proteins? So we can see in this model fungal protein that we study that it's absolutely critical not only to have this polyglutamine track, but also to have the ability to interact with RNA. So when we take out its ability to bind RNA, we also lose this ability for it to form condensates. So what is RNA doing, and why is it so intimately tied to the assembly of these structures, both in health and in disease? So we've done some experiments to show that, in fact, this protein won't do anything on its own without RNA. But as soon as we add the RNAs that it normally interacts with in the cell, we see these beautiful droplets. But it's not the case if we add just something else like DNA or non-specific RNA. So this is telling us that the RNA is not just providing a bunch of negative charge. It's a very negatively charged molecule. It could just be providing a polymer of some sort, but it's not. There's something specific about the RNA. There's something about the content of the RNA that matters. And so we combined this thinking with also sort of a puzzle in our head we had about this protein, which is that we have one protein that we know is important for a whole bunch of different things in these cells. I showed you the branching effect. It's also controlling and localized around the nuclei and controlling their division process. And so it led us to think, well, perhaps there's actually different pools of droplets, that not all the droplets made out of this protein are created equal. Maybe they're not all the same. And so perhaps there's nuclear ones that I'm showing here in purple, but there's also ones that are important for growth. And so we started to test this, and we essentially noticed that indeed, none of the RNAs that drive condensation are ever found in the same spot in the cell. They always seem to be in distinct locations. And that's when we localize individual molecules of the RNA with growth RNAs and division RNAs. They're never together. And then we did a really amazing experiment in a test tube where we mixed these two different RNAs that in the cell are in different spots with this protein, and we saw even in a test tube, they never got together. They were always condensed into distinct, non-overlapping droplets, telling us there's something in that RNA sequence that is essentially giving information to the identity of the droplets. And it made us hypothesize that RNA, information in the RNA sequence, might actually be encoding the droplet identity. And that perhaps RNA actually has a lot of control over who's allowed in or out of these molecular condensates. And so we speculated that there's some sort of sequence information in the RNA that determines who's allowed to be present in a droplet. And I should say this is a problem we don't understand for any of the droplets that have been observed in cells, is who's allowed in and who is kept out. Where does specificity and identity arise? And what we've been able to recently find is that indeed the RNA sequence matters, and it matters for a really interesting reason. It matters because it's determining actually the shape or the structure of the RNA molecules themselves. And so depending on the structure of the RNA, this is actually allowing for RNA-RNA interactions that are very specific and they're sequence encoded. And that's determining which RNAs get sorted into the same droplet, same different droplets, and which ones are actually excluded from each other's droplets. And so this is a really neat function for the RNA code that's not encoding protein, but it's actually encoding the contents of these liquid-liquid phase-separated droplets. So we were very excited to be able to find really the first evidence for molecular identity in these biomolecular condensates taking advantage of this simple fungal system. And because RNAs are sorted into droplets in these many, many contexts, we hope and we think that this will be a mechanism that's widely used in cells. So specific RNAs end up packaged into granules to be transported together in neurons. Specific RNAs end up condensing together under stress. And of course, in development, specific RNAs end up partitioned so that they can give rise to specific parts of the body. And so what are the advantages we found by working in this simple system? We have powerful genetics that give rise to an ability to look at phenotypes. We can essentially quantify loss of function in a system that's very reliant on these with very, very simple and clear phenotypes. The other thing that's really wonderful about working with an organism like this is that there's wild isolates present that have natural genetic variation. These are free-living organisms. And so we've just started a project that takes advantage of actually looking at natural variation in the fungus over many different locations from the Caribbean up through the upper Midwest. And what we're doing is looking for natural genetic variation to study how the sticky patches, how polyglutamine tracks or disordered sequences are actually evolving in relation to aspects such as temperature or climate that might be a source of selection on these compartments that are somewhat at the mercy of fluctuations in the environment. And we already can see some interesting associations between the polyglutamine track length and actually the average temperature at which we are able to isolate the fungi. And then the other thing that we've been able to show is that the droplet properties that we see in fungi are very similar to more complex systems. And so these mechanisms that we can readily identify, we hope and believe are going to be conserved. And so in my lab, we're testing this in a couple of other non-fungal systems that are also really big cells that are syncytia that have lots of nuclei. And specifically, we're looking in developing skeletal muscle, or myotubes, and also the syncytial trophoblast of placenta. And so here I'm showing more green and purple dots to represent compartments of the cytoplasm in these bigger but more complex cells. And what we found is that a protein called Fragile X-related protein 1, FXR1, actually has a very interestingly developmentally controlled splicing, meaning it's essentially made in different forms such that through development, it becomes more and more and more disordered and sticky, suggesting that in development, the process of condensation or phase separation might be being naturally controlled. And indeed, we can see that FXR1 is forming condensates in the vicinity of nuclei, just like we saw with We3, the fungal protein, in this case in the developing muscle. And so what I want to close with is this sort of difficult notion that this new form of cytoplasmic organization that's emerged really just in the last 10 years involves a very tricky balance of these compartments between different physical states. So this transition between a liquid and a solid in some cases can really be the tipping point between health and disease. Not in all cases. There are certainly compartments in the cell that are solid, that are useful and functional. So I don't want you to think all solids are bad, but many of them do cause diseases in neurons. So the challenge here, though, is that this is a normal function, and that these normal functioning compartments actually require the same mechanisms of protein interactions that ultimately underlie the disease. So the question is how can we understand essentially how cells maintain these compartments in these fluid, dynamic states where they're functioning and avoid this transition? And in the case of a situation where this transition has occurred, how can we understand how it might be reversed? So I just want to conclude showing these two different cells from these two different kinds of organisms side by side. And what I hope you can appreciate is that these mechanisms of cell organization that we've been studying in fungi very much arise as a consequence of the architecture of the cell, and that the comparisons between a highly branched network of cells that require as part of their function the ability to transmit information over long distances can serve as a very powerful but simple model of the more complex cells that populate our brains. So I am going to conclude here with just an homage to this place. I have had so many discoveries here, so many moments of exhilaration. I really cannot imagine life as a scientist without the labs here, without the colleagues here, and with all of the fertilization that has happened in this room and on these streets. And I'm completely indebted to my life as a scientist here at the MBL. So I want to just conclude with thanking everyone who's contributed to my work, my former lab members. These are my current lab members, my collaborators on this condensation work, my funding sources. I want to thank the American people for public support of basic science and fundamental discovery. And I want to thank, yay. And I also want to thank my MBL collaborators. And I really didn't have a chance to talk about these projects that I have worked closely with Rudolf and Tomomi. I have a project with Chris Field this summer on fungi from the marine environment. So stay tuned for that. And Hari Shroff. And finally, I'm very indebted to my time in the community of the Summer Institute studying phase transition. So thank you for your attention.
So we do have time for a few questions. Please just raise your hand and just wait until we get to you with the mic. And
hello. That was a great talk. Thank you. I'm just wondering, when you see the solid aggregates in the fungi, are there any kind of pathologies or effects associated with them, with the
Yeah, so we can induce them in, what I showed you was actually in a test tube, but we can induce them in the cell with mutations, interestingly, that are in the protein that can't bind to the RNA. And so while in the test tube, that protein doesn't phase separate, in the cell, when it can't bind to the RNA, it forms aggregates, and it affects the branching and it affects the cell cycle. So we see a loss of function of the protein when it aggregates in the fungus.
It was very interesting. I was wondering if you could tell us a little more about how you think this works in Alzheimer's disease. So the tau hyperphosphorylation that happens in the locus ceruleus first, it happens first in one part of the brain. What is the actual change that would lead to this more change in structure that would form these aggregates, and why would it be happening in one particular place?
Yeah, so I think in terms of why it happens in a particular location, I can't really speak to, but at least in the study that's just come out, it's the sort of phosphorylated form of tau that has this higher propensity to undergo both the phase separation to a liquid, but then also the phase separation to the solid. In terms of why it spreads throughout the brain, I mean, I think there's sort of some interesting thoughts on sort of the prion nature of some of this, that you can actually convert proteins from sort of a more soluble state to this more ordered, aggregated state, and that can happen, and that's one reason you sort of see a spread across the brain. But that's a little bit beyond my expertise.
Demonstration of a range of stickiness within a cell. Should we think of this crystallization as stickiness gone too far, or when is crystallization and phase transition to solid form actually a good thing for the cell?
So it's all a matter of context, right? And so I think it's we to be very careful to not say that crystallization of solid is bad. I think there are examples where it's clearly bad, but there's other examples where it will be a functional form, and it really will be dependent on the process, the biology that's using this. It's all going to be a continuum, and so I think I can't say when. It's really going to be up to the process at play. Mm-hmm.
So there's some evidence, some concern that anesthetic agents, getting a general anesthetic, might cause postoperative cognitive dysfunction. Has anyone looked at the effect of anesthetics on going from that liquid to that solid form?
Oh, that's interesting. Not that I know of. Are there any anesthesiologists in the house? Well, we should work on it. I don't know. That's a good question. Not that I know of. I mean, I have anybody? You have any? I don't know if any of my colleagues have never heard of anything with that. Yeah, it's an interesting idea.
Thank you for your talk. I had a quick question about autophagy or cellular clearance. Like one of the common signs in all of these neurodegenerative diseases is that autophagy becomes impaired over time. And I was wondering if there's anything in your studies that could help elucidate the mechanisms of that autophagy impairment that we see in all of these neurodegenerative
That's interesting,
diseases.
yeah. So I don't know of a direct connection there yet, but my suspicion is that there will be work done on that, but I don't know right now of a direct connection on that, but that's a really interesting avenue to think about. It's a great question.
people who have the Huntington's mutation don't develop the disease until well into adulthood. Would you comment on what prevents the aggregates from forming during their first 30 or 40 years?
Yeah, so this is probably coming from the fact that there are multiple cellular processes that are sort of helping combat aggregation. And so these are the hypotheses right now is that these may be small machines, so machines that use ATP like chaperones and helicases, that if they see an aggregate, they can sort of disentangle it using energy. But that as cells age and they either accumulate oxidative damage or they accumulate stress that might affect their ATP production, that potentially these forces that are helping to keep things fluid and sort of more soluble, that they become less and less functional. And so that this is why there's sort of an age connection, is a hypothesis, yeah.
So how about dissolving the aggregates by modulating RNA binding? Has anyone tried that?
Sorry, dissolving the aggregates by what?
Modulating RNA binding.
Modulating the RNA binding. Yeah, it's interesting. I know there's a small molecule that, there's searches for small molecules in many groups right now, but I do know of one small molecule that does modulate the RNA binding and can dissolve these aggregates. The challenge there is there's a lot of contexts where you really need RNA to be bound by these proteins. So finding something that is specific and has therapeutic potential is limited by the challenge that there also needs to be some functional interaction. But there are molecules that have that capacity.
Hi, thank you for your talk. It's very enlightening. I'm wondering about any immune response that might be noticed in some of these Alzheimer's and ALS patients. Is there any sense that the immune system is reacting to these aggregates in any way? And is there any indication that treatments of certain kinds are interacting with these aggregates being formed?
Yeah, that's a good question. So I don't know, but it may be the scope, in terms of the immunology connection, that might just be beyond my scope. So I don't know of a direct connection there. But again, this is really, really early new days for conceptualizing this. And so we're really in the first wave of understanding it. And so I think like autophagy, that any immune connection, the future, I'm sure, will intersect, but at this point, I don't know of a strong connection. Yeah. Okay.
Could you speak a little more about how many different proteins and RNAs are in the individual balls?
Great
and whether
question.
they overlap? Or...
Yeah, it's a great question. So this is something many of us are trying to sort out, and that is sort of what's the complexity, the molecular complexity within the components in the cells. So we can recapitulate or reconstitute these with very few components, and they recapitulate certain aspects of the biology. But in few cases where inventories have been made of all the proteins and all the RNAs, this would be in the case of stress, so the stress granules that form. We know that there are thousands of RNAs and many, 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 many proteins in these. So it's actually a much more complex mixture of molecules than simply one or two components. But these are early days because we have to be able to purify them, and because they're liquid, they can sometimes be challenging to purify in an intact form. But there's advancements on this. People are starting to really appreciate the complexity of molecules that are in them. Yeah.
So, please thank Amy one more time.
Not to do anything. All right. Oh, thank you, Rudolf. That was so nice. Okay.
Thank you.